Hello, everyone. I'm Wendy Shelton, host for today's webinar. Welcome to our worldwide premier presentation of a methodology pioneered by our two speakers today, connecting gene expression data with physiological simulation. Tom Paterson of Entelos and Nat Goodman of the Institute for Systems Biology will share their approach. I want to mention that these gentlemen have also generated a number of published materials that cover this material, white papers, and that would be available at a later time. These are our speakers for today. Again, Tom Paterson, CTO and co-founder at Entelos, who leads our research methodologies, and his collaborator, Nathan Goodman, from systems biology. Without further ado, gentlemen, I'd ask you to get started with our webinar. I'll hand it over to you, Tom.
Very good, Wendy. Thank you. So good morning, everyone. So thank you for joining us this morning. This has been a very interesting and long journey for Entelos to be working with the Institute for Systems Biology. We take some very different views at looking at and understanding complex biological systems. And for over a year now, we've been working on this collaboration that really started somewhat open-ended with the intent of how do we marry our two approaches? How do we bring together more of the physiologic top-down view that Entelos has for understanding complex disease states with more of the bottoms-up molecular systems biology that ISB is more involved in. And we're really, really pleased today to be able to talk to you about the work that we've done and a particular case study that we've executed that I think is a real landmark in being able to integrate gene expression data with the type of work that we do at Entelos. So let me hand it off to Nat, and he can chat a bit about what the motivation was for ISB to start working with Entelos. Nat, it's all yours.
Okay, thank you, Tom, and thank you, Wendy. Good morning, everyone. Also, so I'm Nat Goodman from the Institute for Systems Biology. I'm a computational scientist working in Lee Hood's laboratory. I'm going to start with something completely obvious, I'm sure, to everyone on the line, so please bear with me. Everything in life sciences is a system, from the molecular systems that occur inside of cells to the physiologic systems that occur in the body at large. Everything that any of us study, they're all systems. So in a very real sense, everyone who works in life sciences is a systems biologist. The really distinguishing, or the characteristic that distinguishes those who call themselves systems biologists from everyone else who do important work in this field is that we, systems biologists, we embrace the complexity of systems. We use the complexity as a tool to help us really understand. We use it to our advantage to understand the underpinnings of whatever biological phenomenon we're studying. The motivation for working with Entelos was that we, and I'm sure most of you fully appreciate that to understand the molecular basis of disease, we need to connect the molecular systems that occur inside of cells primarily. We need to connect these systems to the physiologic systems occurring at the level of the entire body and that are the most direct drivers of disease, disease and health. And so we need a way to go from molecular systems to physiologic systems. Now, we at ISB are really good at generating data, molecular data, creating models to explain what's going on in molecular systems inside cells. That's what we really do very well. And we recognize Entelos as being really good at creating models that explain what's going on in terms of physiologic models. So the goal of this collaboration was to learn how to integrate what we do at ISB with what Entelos does. We can move to the next slide. We're in the midst of a revolution in life sciences. I imagine some of you are old enough to remember when the Human Genome Project was a heretical adventure, when it was considered the work of the devil and had no chance of success, a fishing expedition in a sterile pond, a waste of very limited resources that could better be applied to doing other kinds of research. Way back then, in the prehistory of our field, the task of cloning a gene was a big deal, worth a PhD thesis at least. That revolution continues as we are able to produce larger and larger data sets of many different kinds of data from model systems, model organisms, cellular systems and such. But increasingly, and I think of great excitement, we're able to collect large amounts of data about individual people, whether it's whole genome sequences or molecular data from patient-derived iPS cells and cells derived therefrom, and so forth. We're in an era in which biology is moving from being a data-poor field to being a data-rich science. And it's this transition, this revolution that we're living through right now that gives rise to the need and the ability to carry out the kind of modeling, the studies of systems that we're here to talk to you. Today. And so, as we move through this period of time, and it's happening very fast, and I'm sure all of you are well aware of this, as we move from the era in which studying one pathway in one kind of cell, we're moving from the era in which that kind of research is really the cutting edge to an era in which studying whole systems, whether at the molecular level or at other levels, becomes the norm. And this is the driving force for the collaboration between ISB and Entelos. So let me pass it back to Tom.
Very good. Thanks, Matt. So let me sort of transition there from to a particular class of problem that we're seeing recurrently today around big data, particularly big data in drug development. So if we look at both the challenge and opportunity of precision medicine, the new name given to by NIH and FDA to what we used to call personalized medicine, but the idea of identifying subpopulations. But the general problem of you have a drug's mechanism of action, you have a diverse patient population. It is certainly hoped by pharmaceutical companies that the new drug will be significantly more efficacious than the current standard of care. What may happen is something that is a little less than what is hoped for, but there are still some individuals within that response that are responding well to the therapy. So the idea around precision medicine is to say, well, how can you identify who those individuals are? Now, one of the key aspects for this is the approach of doing molecular profiling to identify